As a member of the advisory committee of the Institute for Religion and Science at Chestnut Hill College, I would like to welcome you to tonight's event. A particularly warm welcome to the many who are here for the first time. The Institute of Religion and Science was established 7 years ago to promote the constructive engagement of religion and spirituality with science and technology, and to encourage a dialogue that is interfaith, multi-science, and civil. To this end, we sponsor lectures, a reading circle, and other events. Do check our website for resources, particularly for past videos and for our blog. Also, be sure to sign up for our mailing list, which can help you stay aware of upcoming programs. Tonight, we are pleased to welcome Gail Woloschuk and her presentation, Perspectives on Life's Technologies. Gail Woloschuk is a professor of radiation oncology, radiology, cell and molecular biology in the Feinberg School of Medicine at Northwestern University. Gail received her BS in biological sciences from Youngstown State University and a PhD in medical sciences from the University of Toledo Medical College of Ohio. She did her postdoctoral training at the Mayo Clinic and then moved on to Argonne National Laboratory until 2001. She is also Associate Dean of Graduate Student and Postdoctoral Affairs at the Graduate School at Northwestern University. Dr. Woloschuk also has a DMin degree from Pittsburgh Theological Seminary, where her work focused on bioethical issues. She teaches science and religion classes at the Lutheran School of Theology, Chicago, and Pittsburgh Theological Seminary. Without further delay, let us welcome Dr. Gail Woloschuk.
So I put my email address up so that if anybody has any questions afterwards, wants to get in touch with me, I'm happy to talk, to chit chat, to email back and forth. First, let me start with my introduction. So I always, I like to start my science religion discussions with this joke, but I have to put the joke on the slide because sometimes I forget the punchline, so it loses its meaning. So the story goes like this. The Pope issued a decree years ago. This is, back in the 1600s, that all the Jews that were located in Rome either had to convert to Christianity or they had to leave. And he had arguments from the Jewish community. And finally, they agreed that they would have a trial debate. So the Jews would pick their best rabbi, put him up against the Pope, and they would debate many different issues. The problem was the rabbi spoke no Italian, the Pope spoke no Yiddish, so they had to do a lot of their debating with hand gestures. So they started out the debate, and the Pope and rabbi sat across from each other, and the Pope raised his hand and showed 3 fingers, and the rabbi raised 1 finger. Next, the Pope waved his finger around his head, and then the rabbi pointed down to the ground, right where he was sitting. Then the Pope brought out a communion wafer and started to eat it with a chalice of wine, and the rabbi pulled out an apple. And with that, the Pope stood up and he said, "That's it, you've won, you've bested me at every point." So the cardinals met later with the Pope and they asked him, "We didn't understand this debate. What happened?" And the Pope said, "First, I held up 3 fingers to represent the Trinity." And he responded by holding up a single finger to tell me that even though there's the Trinity, there's still one common God that we have. Then I waved my finger around my head to show him that God was all around us. And he responded by pointing to the ground, saying he was right here with us. And then finally, I pulled out the wine and the wafer to show that God absolves us of all our sins. And the rabbi pulled out an apple to remind us of the original sin. He bested me at every move. I could not continue. Meanwhile, the Jewish community gathered around the rabbi and asked how he'd won. And the rabbi said, "I have no clue what happened. First, he told me that we had 3 days to get out of Italy, so I gave him the finger. Then he told me that the whole country would be cleared of Jews, and I told him that we were staying right here. and then what happened? And he said, and then he took out his lunch, so I took out mine, and there we go. We can stay. I tell this story because it's one of miscommunication. And I often believe that the science religion dialogue that we have ongoing among us is kind of like the Pope and the rabbi talking to each other. We're talking past each other, we're using hand gestures, but we're often not talking with each other. So I think that that's a good way to think about things. Another approach, and as was mentioned in the introduction, I'm a practicing scientist. I have a research lab, but I also have a Doctor of Ministry degree and work a lot in the science religion community, particularly the Eastern Orthodox community where I'm a member. So there were surveys done a number of years ago, and they asked children, what do you think about scientists? And the children said that scientists speak a foreign language that no one can understand. And that was actually true. I think we do speak a language that nobody can understand. That we live underground, I mean, that one is a bit of a stretch. We wear white coats even when we sleep. We wear glasses and we don't require food. I'm not sure where all these ideas came from, but most of them, are not quite in the realm of accuracy. But it's the view of adults that tells us something. So the adults said, yes, scientists speak a language nobody can understand, but they also said scientists are antisocial and that they've cured diseases that only rich people can get the cures for. And I think that points to the great distrust that we have in society for scientists today. And somehow science needs to work to overcome these kinds of issues, because neither one is true. I mean, most scientists are not antisocial, and certainly we have not cured diseases that only are given, where the cures are only given to a few people. Now, science is important for us. It is about understanding the world around us. Through our education, we learn about all the things that are ongoing in the world. Through medicine, I think there's probably nobody who's listening that hasn't been impacted by medicine, either through some new technique for diagnosis, like the CT scan that Hounsfield won the Nobel Prize for, or the MR, or any of the diagnostic techniques, the treatments that we use. And I work in the cancer field mostly, and we have a lot of treatments that that have come from science itself. Certainly there are quality of life issues and end of life issues that are important. Think about ecology, the dialogue that goes on about recombinant foods. Should we be using recombinant foods? What about environmental issues? And then the topic I want to focus on a lot today, which will dovetail with these others, is about technology. We have new technologies that are shaping what we can do. Most of them are coming from cloning technologies, genome science, beginning of life issues, genetic counseling. And I have to tell you, I'm not sure I'll get through my entire talk in this amount of time, but I'll get as far as I can. And hopefully we can cover some of the other stuff when we come to discussion toward the end. So I want to emphasize that it might not be apparent, but science actually drives the ethics. I could use the COVID example, but I think I'm going to use the HIV example because it's older and more distant from us and easier to see. So when HIV/AIDS first came into existence, nobody knew what AIDS was caused from. They didn't know it was caused by a virus. They didn't know how the virus was transmitted, and it was scary for people, just as COVID was scary for people. And so in the early days, there was talk of quarantining everybody that had AIDS, to restrict their interactions, to restrict their medical care. A lot of times they were left in hospitals with not ever touching a person, as we heard so much with COVID. And then finally, science demonstrated what the route of transmission was for AIDS, that it was caused by the HIV virus, that it was transmitted only by bodily fluids. It could not be spread through the air. And so then it now became unethical to say, we're going to quarantine people, because we knew how it was spread. We knew how to stop it from spreading. We knew what to do in order to change our behaviors to not have it spread. So the facts that we learned from science helped to shape what became an ethical treatment of patients. And I think that hand in hand relationship between science and certainly ethics, particularly for technologies, for diseases, is very important. We all saw it with COVID as well, where people feel a lot better now that we know how it's transmitted. At the time, we were like wiping down every surface that we possibly could with alcohol, staying in our homes to prevent others from getting it. Now we know that masks can protect us. We know the many different things that can be done, and that was because of the science that came about. So just a quick definition of what ethics is. It's a branch of the philosophy, and it involves coming up with a concept of what is right and wrong conduct. It's often used for discussing things that are disputes. It comes from the Greek word ethikos, which means custom or habit. And it investigates the best way for humans to live, what kinds of actions one would consider right or wrong in particular circumstances. Now, there are a lot of people that argue that ethics is not a good way to do things because it's not moral in a sense. And certainly many people in the church argue about whether ethics is appropriate or not. Ethics is something that is done in the broad public context, and it's not necessarily done for morality as much as it is for trying to treat people appropriately. So I have a quote here from one of my favorites. Cyril of Jerusalem wrote in 347, "For the method of godliness consists of these two things, pious doctrines and virtuous practice. And neither are the doctrines acceptable to God apart from good works, nor does God accept the works which are not perfected with pious doctrines. For what profit is it to know well the doctrines concerning God, and yet to be a vile fornicator? And again, what profit is it to be nobly temperate and an impious blasphemer? A more precious possession, therefore, is the knowledge of doctrines. Also, there is need of a wakeful soul, since there are many that make spoil through philosophy and vain deceit." So this was a view of ethics in 347. I think it suits our thinking about ethics today, at least to some extent. We might not in today's world be as comfortable with God doctrines and other perspectives, but that like concept about being lacking deceit and trying to understand through appropriate practice, I think very much suits the things that we talk about today. Okay, so I'm gonna try to get through a couple of new technologies. I don't know that I'll get through all of them, but I'll try to get through a few of them. The first one will be in vitro fertilization. This one's relatively easy and relatively short. So I'm hoping I'll have time to get into gene therapy, for instance, is the next one. So way back in 2002, there was a council that was developed on bioethics, and it was set up by the president at the time, and he impaneled a group of people to talk about what should we do with human cloning and how do we think about human cloning. And what was interesting was that what the committee did as their first task was not to sit down and define cloning and define all these different techniques. They actually read a series of books to define that were novels to talk about what does it mean to be a human being? How do we understand human beings? Before we start thinking about cloning and technology and all these different things, we need to think about what it means to be human. And the list of books is available on the web. It was a very interesting list, and it certainly affected the committee's discussions later. They all said that it was probably a really good way to start with it. Now, probably everybody on this call knows how in vitro fertilization takes place, or how regular fertilization takes place. This is the woman's reproductive tract. Here is the ovary with the oocyte, the egg, popping out at ovulation. It moves down the tract. Eventually, if it becomes fertilized, it goes down, down, down. Eventually, at around day 9 or 10, it plants itself into the wall of the uterus, the uterine wall. And so we have this process that takes place. This is what happens inside the woman's womb. And you can see that it's going from a 1-cell stage to 2 cells to 4 cells to 8 cells in all of this. Well, what people try to do with in vitro fertilization is to take this out of the womb and to put it into a test tube, where in most examples, the embryo can't develop too much more than about day 6 or 8 days. And then it's implanted into the womb of a woman who's been made hormonally pregnant. And hopefully the fertilized egg, the product, the embryo seats itself in the uterine wall and becomes a mature child. And this is just a picture. Actually, this is from a mouse. My lab works with mice, not with people. This is a mouse egg, and you can actually see there's a bit of a shell around it. That's the way it looks in humans as well. There's a bit of a shell around the egg to protect it. And then as it starts to develop, I don't know how well you can see it, but here are a bunch of cells. This is about the 16 cell phase right now in a plate. So this is a single cell, and this is all the cells in a plate, just so you can see what they look like. Okay, so there's this process that happens naturally that we now try to use technology to change. Why do we do in vitro fertilization? And what does it mean? Well, in vitro means in the test tube, and fertilization means exactly the joining of the egg and the sperm. So instead of they're taking place in the womb, as we just looked at, it takes place in a test tube. The clinical definition of what it's used to treat are fertility disorders. But at the end of the day, it's basically to help couples that are not fertile to bear children. Originally, people called them test tube babies because they were born in test tubes, although honestly, they were always born in plates. So they should call them Petri plate babies, but nobody does that. So what we do is what most scientists do is they isolate eggs from the mother, sperm from the father. They mix them in a Petri culture plate. They let them develop for several days, and then they implant them back into the mother, as I said. And the leftover fertilized eggs in the test tube can be used for a variety of purposes. Sometimes they keep them to use for new embryos. Sometimes people talk about using them for embryonic stem cells, although that has not happened. And sometimes they just line our freezer chests, never used at all. So there are many questions that come up in the ethics of discussion of the IVF technology. And I'm going to do my best to not make judgments on them, but I'll talk about the arguments that people make in the course of this. So there's one argument that people use against in vitro fertilization. That is, of course, that it's not natural. But most issues of technology are not natural, including the use of chemo for cancer. The second is what's the idea there about doing IVF? Why can't you just go adopt children? And some people say that it's a selfishness on the part of the parents who insist upon passing their own genes on next generation rather than caring for a child that needs to be raised. There are some that argue justifiably that there are high costs of adoption. It's sometimes difficult to enculturate an adopted child into a household. There are also a lot of issues about you adopt and years later the real parents are looking for the child, or the birth parents are looking for the child. And then finally, IVF is very expensive. I mean, adoption is expensive, but IVF is much more so. So these are arguments people use. I'm not going to try to argue one way or the other. I have friends that have gone through IVF, and they feel very strongly that it was a good thing for them, for their families. Here's what it looks like. And actually, my lab does work not with human embryos. We work with mouse. But you have like a little tip that holds the egg in place. And this was this is actually a fertilized egg. There are few eggs here, so this is an embryo. What this tube is doing is it's sucking out one cell so it can test to see if the embryo is going to be normal. So they're doing genetic testing here, but you use the same kinds of techniques to be able to move around embryos so that you can choose which embryos might be more healthy than others. But there are unique aspects to this technology that need to be mentioned. One of them is what we call pre-implantation genetic diagnosis, where while this embryo is in the test tube, you actually run a test on it for its genetic diseases. And then you decide, whoa, this baby is going to have genetic diseases. We are not going to implant that one in the womb. We are going to implant a different one. There was an example in Minnesota a number of years ago where a child had leukemia at a young age, around 2 years old, and the parents got pregnant with a child who would have the right cell type to be able to replace the leukemic cell in their 2-year-old child. So they were actually selecting for particular proteins that would be made in the embryo to make sure they could correct for the defect in their other child. And then in 2009, for the first time, we see clinics popping up. LA was the first to do this, where they would offer screening for gender and physical traits. Here's one of these advertisements. And the article about it talked about unnatural selection, where you could actually take the eggs that were fertilized with sperm, choose the one that is, remove the cell using that technique we just looked at, test it for female, green eyes and blonde hair, or male, brown eyes and dark hair, or whatever you want, implant it back in the embryo, and there we go. This one is the brown-haired, blue-eyed baby girl. Certainly this goes on frequently. It's been going on since 2009. There's nothing illegal with it, but I do think it pulls some ethical issues into question. There's not just screening for hair color and other traits, but there's screening for, no diseases, lacking particular diseases. And a lot of times, I'll mention this later, but we don't always understand how genes and disease relate in many examples. And so we may be altering our genetic population in ways we don't understand. Now, just so we're clear, this is the idea of twinning. So there can be twins that are born from these in vitro fertilization steps, and usually there are twins. There can sometimes be triplets. Some in the old days they used to have even 6 babies born at the same time, because they would implant in a bunch of embryos and they would all take. We no longer do that because it's not healthy for the mother. But in diamniotic twins, the 2 babies cleave away from each other in days around day 3 at the latest. In those that have a single placenta, but 2 amnions, it's at 8 days. Ones that have 1 placenta and 1 amnion, it's up to 13 days. And this is probably the last point at which you would get kind of normal twinning, is around day 13. And then after that, we start to see versions of conjoined twins, which will lead to many problems that must be handled surgically. Okay, so I deliberately put the in vitro fertilization first so that we can set the stage for stem cells as the next important discussion. So what is stem cell research? It is where we take undeveloped cells and we try to use them as replacement cells in adults. They are put in the body in certain locations, and when they're put in the right location, they mature to be that particular cell type in the body. There are many people hypothesize that there are diseases that could be treated with stem cells, and you've probably heard of some. I recently had knee replacement surgery, but one of my neighbors had stem cells put in her knee to replace her knee joint properly. Now, honestly, it was not very successful for her, and most of that technology has not been so good for the knee. But in cases where there aren't many choices for what you can do, like a spinal cord injury, for instance, people are trying it. There are some side effects that come with it, and one of the side effects is when you put stem cells in, a lot of times they behave a little bit like tumor cells. And so in the example of spinal cord injury, there were some people that developed spinal cords, but developed cancers in the spinal cord. But there are lots of potential uses of stem cells, certainly for heart attacks, for muscular dystrophy, for wound healing, bone marrow transplantation. In fact, we use bone marrow transplantation now. We use stem cells. We've been using it for decades. It's a form of stem cell transplant that we accept is the bone marrow transplant. So the issue then becomes, what kinds of stem cells do we use? The adult stem cells have been used for decades. As I said before, they've been bone marrow transplants where we look for people that match for their proteins, and we give the bone marrow from one to the other. My own church has supported their use, and so have most churches have not been opposed to adult stem cell therapies. There are a few that have. They've been developed for cardiac diseases with some limited success. There are new approaches where they take adult cells and they reprogram them to be stem cells. So they make them go backwards in their function, and then therefore they can serve many different functions. But the problem with adult stem cells is that, again, just like with bone marrow stem cells, you must have a match. So the easiest thing is going to be to take my own stem cells and use them to treat me. And that's what people are trying to do now. There is an older method, or let me put this in a more controversial method, that was called embryonic stem cells, where they took those in vitro fertilized eggs or embryos, and they would use those as the stem cells. They were less limiting because they can develop into many different cell types, but the problem was that they have to match from person to person. But this was an example where the religious communities actually had an influence over the discussion, because many groups, including Protestants and Catholics, did not support the use of IVF embryonic stem cells. They opposed it, and scientists actually said, well, this isn't going to work. We're not going to get enough support to do this. We better go back and develop new methods for adult stem cells. So it is one of the rare examples where the religious community has actually changed the shape of how science is done in the modern age. This is sort of the concept here with the embryonic stem cells. Is you would take them out of the human fetus. You would then allow these cells, you'd implant them into circulatory system or nervous system, immune system. And when they are in that environment, they become that cell type. We don't really understand why, but it happens. And they now can make up for deficiencies that you might find in those particular systems. And of course, Alzheimer's has been one of those that people have been looking for a long time and haven't had, good luck. And so then to add to our concept of IVF, leftover fertilized eggs and test tubes can be used for embryonic stem cells. And that would be the concept. But as I said, it was met with so much resistance. It's not really even approved for most uses today. And in fact, most groups that favor stem cell research do not support the use of aborted fetuses for those purposes. There are some groups that do support using in vitro fertilized eggs for that purposes, because they're sitting in freezers, they're not being used, they've never been implanted into a woman or into a womb. Nevertheless, even those, there is some concern about their usage. So what then happened was new techniques were developed for inducing pluripotent stem cells. I'm not going to go into what that means. I just want you to know that's what this abbreviation comes from. But you would get mature cells. You would put a virus in that has genes in the virus that make them become more stem-like. You would then have these cells that originally came from mature cells that are now more stem-like cells. And those are cells where they can come out of any person and can be used for that purpose. There are pros and cons of using these induced pluripotent stem cells. You do have to take the adult stem cells and reprogram them. They don't happen very efficiently. It happens with a very low rate. When you do it, you have to put in this viral sequence, this virus. This virus puts itself into the genome and it inserts itself into the DNA, into the genetic material of the original cells. And whenever that happens, that comes with a risk of coming in near a gene that causes cancer and maybe causing cancer. And that became a risk that maybe was a little bit too great. They can't even on their own go on to become tumor cells. And sometimes the reprogramming is incomplete. But the advantage was you don't need embryonic cells. You don't need to worry about graft rejection. But now we've moved from using stem cells to moving gene editing technologies. And I'll mention those in the next few moments. But there's a lot of work being done with developing artificial organs. Here's an artificial heart that's being generated. So we will see a lot of that. And finally, we come to my third technology here, which is gene editing. And I believe that this is the technology du jour. My lab does a lot of this. We do it again in mice, not in people. But I think there are some questions to think about here. The first question is, should we do gene editing? And the answer is we already do it. The next question is, should we do it in humans? And the answer is we already do it. The third question is, should we do it in eggs and sperm cells? Should we do it in those germ cells, not the ones that make up our body, but the eggs and sperm that will pass it on to offspring? We don't already do it there, and that's controversial. What do we use to justify using gene editing? And what influences do we need to consider? So let's look at the examples. We already do it. Here's the example of how we already do it. Lab animals, we do it all the time. We've been doing gene editing for probably 6, 7, 8 years in lab animals. We're trying to understand how particular genes are associated with disease. And so we edit the gene in order to find out how it's associated with disease. But we also use it as a tool for material production. So think about insulin for a moment. People used to inject pig insulin into themselves. And what happened? They would get a terrible immune reaction to the pig insulin after a while. So we needed to come up with a way to make lots and lots of human insulin. So we took the human insulin gene, we engineered it into the mammary gland of a cow. So when the cow made milk, it also made insulin. And then we milk the cow and we purify the insulin. And that's how most of our insulin is made today. And it's from this gene editing. Certainly, we engineer a lot of organisms to make them be resistant to certain stresses. And then there's the issue of genetically modified crops, which is a very big issue in both Canada and Europe, and I would say some in the US as well. Here's just an example. The first genetically modified mice that were done. These mice had put in them a protein that makes them glow in the dark, or makes certain organs glow in the dark. Here's the normal animal. Here are the 2 mutant animals. You can see how it's glowing. It worked really well. Should we do it in humans? And the answer is we do. When we do it in humans, we do it only for somatic cells. So I mentioned this earlier, I'll say it again. We have 2 kinds of cells in our body, eggs and sperm that we call germ cells, and somatic cells, which is every other cell in our body. If we modify the somatic cells in the body, it's never going to be passed on to offspring. But if we modify eggs and sperm, it's going to go on to their offspring and their offspring and their offspring and their offspring. So what are we going to do? By modifying eggs and sperm, in a way, we're modifying all of humanity. We're putting new genes in the human gene pool, as we call it, or the pool of the entire genes of the human genome, and therefore we're causing modifications that could create problems. Which are the ones we're doing today in the somatic cells? Most of them are genetic diseases that are single gene deficiency diseases. I will tell you that is not the most common type of disease in humans. Most of our diseases are multigenic. Cancer is multigenic, diabetes is multigenic, but there are a few that we know are single gene deficiencies. I'll mention one in a moment. So most techniques have involved gene replacement, not modification, but there is some work being done on modification. And there are some older technologies in gene editing before the development of a modern technique called CRISPR that I'll mention in just a moment. But here's the story of some of the first genetically modified children. So there's a disease called severe combined immunodeficiency. It's present in children. They inherit it generally from their mothers, and they don't have a functional immune system. At the end, they're missing only one protein. So they would raise these children in these so-called bubbles. They would call them bubble babies because they would grow in a bubble. They would never have any contact with the outside world because as soon as they did, they would get infected and die from it. But it was learned that what the gene was that was involved, we created a virus that you could infect in these children. And now they could make the protein they were missing, and they could come out and live a relatively normal life. They had to be reinfected with the virus like every 10 years, but this was a genetically modified human that was genetically modified. And as I said, really, this is one of the first examples. There are other examples. Should we do it in the human germ cells? And I think there are three main questions that need to be asked, and I don't have the answers to this. I do not favor doing this right now. I think that we need to have a lot more discussion on it. But number one is that all the existing techniques that were used were limited. Remember what I mentioned about the virus. You infect, and it could put itself anywhere in the genome, and when it did that, it could cause cancer. Well, there's a new technique called CRISPR that I'll show you in a moment. CRISPR takes away that problem. I still have the worry about damage to the human gene pool. And then I think there are ethical concerns about who do we modify and who don't we? So there's this new technique that came out maybe 6-7 years ago. It's called CRISPR. CRISPR is a protein nucleic acid complex. What does that mean? It is a protein bound to DNA or RNA. It's a protein that's bound to genetic material, but it's usually a very small piece of the genetic material. And what it can do that nothing else that we know of can do is inside our genome, inside our cells, inside our nucleus, we have billions of possible places for a DNA to be cut. But CRISPR allows for a very precise place to be cut. One place in all those billions of possible places can be targeted and designed to be cut with CRISPR. This is amazing. It changed the way we do everything. This is a picture of what it looks like. Here's the CRISPR itself, the protein. Here is the DNA that is inside of it, and it's got an RNA in that helps to guide it. I'll show you how this works in a moment. But this is the structure of what this looks like, and this is how it works. So what we do is we put in a piece of DNA that we want to put into the genome. We put in what we call a guide RNA, which takes our CRISPR, we call it CRISPR-Cas9 often because it's got another protein with it. But you put this together with the guide RNA. The guide RNA is also a nucleic acid, and it carries this CRISPR-Cas9 into the genome to exactly the place that you want it to. It can cut out the DNA and put in the new DNA that is the right sequence. So for the first time, we can cut specific locations in the human genome and replace it with a new gene. So think about those bubble babies. They can't make this gene anymore. Well, instead of creating a virus to do it, where the virus could put itself down anywhere in the DNA, now we put the correct gene here. We put the guide RNA that takes it to the right gene, bind it into the CRISPR-Cas9, goes into the nucleus, cuts out the bad gene, puts in the good gene. This can be done with great efficiency and with almost no errors. So this is going to revolutionize what we think about. It has improved accuracy. And the one thing I didn't mention is it actually has a tool where we can shut it off. So we can turn it on and we can turn it off when we want. Now, I have about 5 minutes or 7 minutes. I won't go into all the little specifics of how this works. If anybody wants me to, I will. But I will say that this was discovered actually in bacterial systems. So bacteria were using this as a defense against viruses. They were cutting out viral sequences and protecting themselves in that way. We introduced these sequences artificially, but our own body uses normal processes to make it go. It is the most efficient, easy, flexible, and it's very, very cheap. We can do it in the lab in a very short time. The so-called off-target effects are minimal. What does that mean? It means that we get the gene we want and we don't get any other genes in the genome. So therefore it's highly accurate in what it does. But here's my worry. So what is the human gene pool? A gene pool, it's kind of like what you think about. What's a swimming pool, okay? A swimming pool has all these people floating around, swimming around in it. A gene pool has all the genes floating around, swimming in it. And in most examples, a human gene pool contains all the genes of all the reproductively active or capable people in the world at any one time. The mouse gene pool contains all the genes of any reproductively active or capable mice in the entire world. That gene pool now it's going to be changed if we start fooling around with genes that are in eggs and sperm, because we will be making differences in the gene pool, and we don't really know what differences those will have or what consequences those will have long term. Gene pools can change naturally. Certainly survival in particular environments, natural selection against certain processes changes our gene pool. The gene pool of 5 years ago was not the same gene pool as today's. The changes in gene frequencies will vary from in one way to another. But it's not clear to me who decides on what cells will happen. So I want you to think for a moment about how we decide when a process is ethical. In hospitals, we have a group called the Institutional Review Board. And what the Institutional Review Board does is they decide whether this technique is going to benefit this person or is it going to harm this person. But who decides whether a particular technique is going to hurt or benefit humanity? Nobody. And the other concern is that most of our studies, they last 5 years because that's how our funding works. But if you want to follow problems with the gene pool, you're going to need to watch changes over generations, not just for a very short time. And I do think that there is some hubris that comes in where we claim that we can predict the impact of muddling around with the human gene pool. For a lot of our genes, we know only a very small amount of their function. And of course, as we progress, we learn more about the functions of different genes. So if we think that we can predict by making this modification, it's going to have this impact, I promise you, we don't know that for many of the genes that we will work around. It might be possible to do this with single gene traits like that bubble baby situation that I mentioned earlier, but for more complicated diseases like diabetes or schizophrenia has 120 genes associated with it. I mean, how do you think about that? Now, I don't think I have time to talk about sickle cell anemia, even though I would love to talk about it. But I want to make this one point, which is that genes can be good or bad depending on the tissue, the time when they're active. For multi-gene traits, they have different influences that we can't predict. I do believe, though, at the end of the day, the goal here is to eliminate human suffering. There are diseases like Tay-Sachs disease and Huntington's disease that occur with, serious neurocognitive concerns, horrible diseases. If we could cure those, and those are diseases where it's not likely that we're going to be able to fix them by a single gene. We're actually going to have to make changes to multiple plate parts of the body. Stem cells are not going to do it there. It's a problem. Alternatively, there's probably some suffering that is needed, but needless suffering, I think, is something we don't want to have. And then, economics treating most of these diseases is going to be very expensive. Here's a guy I know who developed a do-it-yourself CRISPR kit. You can buy it online for $150. He's selling it out of his garage in Palo Alto, close to Stanford. I don't know what you would CRISPRize with it, but okay. I think I said all this. Okay, I'm not going to go through the end of life. I just don't have time enough to do it, but I'll make a few concluding thoughts. First of all, biotechnologies increase the number of decisions and turning points in our lives. Years and years ago, if a couple couldn't have a baby, they didn't think about IVF. Now they've got to think about, do we adopt or do we do IVF? Do we have a surrogate mother? Do we think about choosing the hair color of our baby? I mean, so questions are very different. And I think these leave challenges for families, but particularly for people in religious communities to try to deal with them. I strongly support collaboration between pastors and health professionals. And I believe that it's in religion science dialogue like this that we end up helping people come to some of these conclusions. I'm gonna end on one. I'll end on this point about unity in science and religion. I think that there are common themes in religion and science, and technology fits as part of that science component. First of all, both have a belief in mystery, that there are things that are unknown and maybe even things that are unknowable. Both accept that there may be more to the story than they can explain. Both have a mentorship model, at least in many religious traditions. There's a spiritual director who provides advice to and counseling to members of a parish community or a congregation. And yet in the scientific director from my students, I mentor them. I mean, and I don't teach, I mentor them. I'm trying to train them to be good scientists. The test of success is not an exam, it's living life and it's doing the task at hand. And with that, I'm going to just end. And thank you very much. And I think I'm only a minute over. Hey, there we go. Let me start my share.
That's excellent. Gail, that was riveting. I myself teach a science and religion class with I'm a theologian and a philosopher, and I teach with a chemist. And one of the hardest things that we find is involved in teaching a science and religion class is to stay balanced between science and religion without tipping off into one or the other. And your talk just now was, you went back and forth across both domains with incredible skill, and you seem to do it almost effortlessly. I was riveted the entire time. So I think that's my pleasure, sincerely.